Thank you. So as I'm sure we're all well aware, introgression and incomplete lineage sorting can result in gene trees that disagree with the species tree. And the network multi-species coalescent model predicts the frequency of the 3 gene tree topologies, or the concordance factor, based on the branch length in the species tree and coalescent units and the extent of introgression. So the distribution of these tree topologies provides a wealth of information about population processes. And in the context of 3 species, we can easily estimate this distribution by first identifying one-to-one orthologs, constructing gene trees, and then counting the 3 topologies. But what if we wish to consider a larger set of species? A common approach is to reduce this problem to size 3 by subsampling rooted triplets that probe each species tree edge. However, any integration along this path shown here will result in the same tree distribution regardless of the species involved. We can mitigate this problem by sampling multiple triplets. However, combining information is tricky, and the number of triplets can grow exponentially with K. So our work here is we're proposing a new solution where we reduce the problem to size 3 in a different way by examining each internode separately. As we can see, each internode induces a smaller tree with 3 leaves and 3 branching orders. So for internode UV, we can consider the topologies where either X branches first or Y or Z branches first. So given a phylogenomics data set, how can we identify the support for each branching order? So the way we do this is to first find the area of the gene tree that corresponds to this branch in the species tree. And we do this by using reconciliation to identify the species mapping of each gene tree node, and then extract this part of the gene tree and ask if it supports one of the 3 topologies. And we can assess support for topologies by considering edge UGVG, and the gene tree supports one of the 3 topologies of US, VS, if and only if exactly one of these conditions is true. So in the first case, the leaves of the species mapped to gene tree node ZG is a subset of the leaves of ZS, and then the union of the species mapped to XG and YG is a subset of XS and YS. And similarly, we can consider the other 2 topologies where we can see ZG, the node mapped to ZG is a subset of the leaves of XS, and then the leaves of YS. So what is the rationale behind this set notation? Well, the 3 topologies are different bipartitions on the left and right children of U. So the descendants of the species with the left child of gene UG should inherit the descendants of the left gene, and similarly for the right child. With some extra bookkeeping, which we don't have time for right now, we can construct these sets and then account for incongruence due to ILS, gene duplication, or loss. So we're going to look at some examples to get some intuition. So in this case, ZG maps to XS, and the union of XG and YG, which is here GB and GC, map to the union of YS and ZS. And so edge UGVG satisfies condition 2 and counts towards the topology with XS as the outgroup. In this case, we have ZG with children mapped to A and B and maps to VS. VG has children mapped to A and C. And as we can see, there's this set overlap, and so none of the bipartitions is satisfied in this case. And so UGVG does not support any of these conditions, and this could be due to either duplication or distant integration. So by using these set mappings, the 3 conditions can include incongruence due to paralogy, and our method can be used with multi-gene family trees. As an example, here we have a duplication that occurred in the ancestral species US. And as in the previous as in the previous example, the edges descending from that duplication do not satisfy any of the bipartitions, but both of the subfamilies do. So the blue family satisfies condition 1, the yellow family satisfies condition 2, and they can each count votes for those bipartitions. So in our method, for every species tree branch and every gene tree branch that maps to US, we perform this test and record the support in a single traversal of the species tree. Our approach has been implemented into the No-Tongue software in a version called iTriplets, and we're still doing a lot of testing, but we're going to soon be looking for beta users and testers. But there are some limitations to this approach. The first is that it assumes ILS does not extend across multiple adjacent branches and may not be suitable for massive radiation events. And currently, it only recognizes nearest neighbor integressions. It filters out those distant integressions, so integressions between leaves that are not nearest neighbors. And we'll probe this limitation with some simulations, which I'll show you now, or in a moment. So that brings us to our next section. We analyze data from simulations and a case study in the literature to assess how this approach works and probe its behavior when there are multiple short branches or a combination of integression and ILS. So for simulations, we simulated 1,000 gene trees using the Phylogenetics simulations. And these were based on this species tree with 25 leaves. And we set all branches to a length of 3 coalescent units, and then we varied a number of characteristics and analyzed behavior. To assess performance, we looked to see if iTriplets recovers the correct branching order frequencies. So we asked if the observed frequencies are statistically indistinguishable from the simulated frequencies. And then we asked how accurate are these frequencies on estimating branch length and hybridization rates. And we got these from the multi-species coalescent equations. So in our first set of simulations, we considered ILS on 2 short branches. So we first made this branch in the species tree short under 4 different levels, and then we added a second branch in various positions relative to this one, and considered 7 different combinations of branch lengths. When looking at when short branches were adjacent, the distributions for only 3 of those edges were statistically different from the simulated, and no distributions were significantly different for all other edges. So that's great. In real data, we wouldn't be able to compare observed to the truth, so we also wanted to ask if the frequencies are significantly different from minority symmetry expected under the MSC. And in all cases, minority symmetry was preserved, even though some of those frequencies were perturbed. Looking at estimating branch lengths, we considered the error, and you see the error here in this histogram. And we performed pretty well with an average error of 0.07, and the exceptions here were in those cases where the 2 adjacent branches were both short. We also considered nearest neighbor hybridization and whether we can distinguish introgression from ILS. So in this case, we added a nearest neighbor hybridization event under 2 different rates of integration. And then we added a short branch in various positions relative to this hybridization. When comparing observed frequencies to the expected simulated frequencies, there were 12 distributions that were significantly different, and 11 of these were in cases where there was extensive introgression, and they were all on the branch. 8 of them were on the branch highlighted in yellow. So this kind of indicates where we might have some difficulty parsing out this information. If we look to compare the observed frequencies to those expected under the MSC, so no integression, or sorry, no ILS, yes, no integression. Only those distributions associated with the hybridization event were identified as statistically different. So this indicates that these statistics we extract can be used to distinguish edges with suspected integration from those with incongruence from ILS alone. As before, we considered error between inferred and true branch length, and again, performed fairly well with an average error of 0.07. And when looking at error between the inferred and true level of integration, we do very, very well with an average of 0.01 error. So simulations did not include duplication and loss events, where this approach might be very helpful compared to other approaches that are currently available. And so we assessed performance on real data. We considered a case from the literature. So this was based on, we took this case from Pollard et al., where the authors were interested in assessing ILS in the Drosophila melanogaster group, specifically melanogaster, yacuba, and erecta. And looking at single copy orthologs, the resulting distribution they saw displayed minority symmetry. And they methodically considered lots of other evidence to affirm that the observed incongruence is due to ILS alone. So it makes an excellent benchmark for testing methods designed to detect ILS. So to assess if internode triplets can recapitulate this observation with multi-gene families, so instead of single copy orthologs, we looked at a wider set of 12 Drosophila species and 13,000 trees for multi-gene families that ranged in size from 3 to 144 genes. And we were able to calculate all topological distributions in under 2 minutes. And when examining the same edge as Pollard et al., we observed a striking similarity in the concordance. In addition, the frequencies of discordance were more symmetric. We can also look at which branches statistically differ from minority symmetry and perform other analyses downstream, which we don't have time to discuss today. I just want to leave you with the note that the method is fast and can process a large number of gene trees or a large species tree relatively quickly. Is that, Okay. So in summary, our method reproduced results from simulation, eliminates noise due to paralogy and missing data, reproduces results from that benchmark study by Pollard et al., and can support clade-level analyses by assessing congruence on every internal branch. We have lots of future work to do, including more extensive simulations, especially those that include duplication and loss, as well as much more algorithmic work and software implementation, such as extracting branch lengths associated with each topology. And I'd like to thank current and past members of the Duran Lab who have worked on tree comparisons and the no-tongue software. Thank you.
Hello, everyone. This is John work with Cecilia Ne, John Fogg, John Rhodes, and Elizabeth Alman, all great people. So some preliminary concepts. So we have a rooted network, the thing we use to represent evolutionary histories between species in the presence of hybridization. So at the top we have the root. All the edges are directed away from the root, and the red edges represent our hybridization events. Now here you see a node labeled X. That is our lowest stable ancestor, which if I go from the root following the direction of the edges to any of the taxa, I will have to pass through X. And is the lowest node that has that property, okay? So the LSA network is the network obtained after removing everything above the LSA. So we just remove everything above. It's also a directed network. Everything is directed away from the LSA. And the unrooted network is obtained after ignoring the LSA and then undirecting all the black edges, but the red ones still are oriented. Now, this unrooted network can be obtained in several ways from different LSA networks. The LSA can be placed in different places, can be put in different places, but for example, I would not be able to put it in the terminal edge of B because that will be inconsistent because that hybridization event will be contradicting. So the basis of this work is a multi-species coalescent model that It's a stochastic process of gene tree generation in the presence of hybridization and ILS. So here we see just a discrete representation, but we're in the continuous one. Okay, so following Claudio Celis Lemos and Cecilia Ney, we looked at concordance factors, which concordance factors is given a network, you look at 4 species at a time, and for these 4 species, you look at the probability under the coalescent model that it will display each of these quartet trees. Okay? These are relevant because several inference methods have focused on concordance factors like SNAC and ANUC and TNIC. Okay, our first result is that we prove is, suppose you have 2 rooted networks, metric rooted networks. If when you unrooted them, they have the same unrooted network, then the set of concordance factors will be the same. What this is telling us is that if you're using concordance factors, you cannot distinguish the root. Or you cannot also know anything about the LSA. So if you have a complex hybridization happening above the LSA, there's nothing you can do about when working with concordance factors. So I have to talk about 2 sublobes. These are really technical to define, so I'm going to be super informal. You know when you see them, but to formally tell what they are is like really tricky. So in a network, just look at the things that have like these 2 boundary nodes, and everything that is happening inside them. So in this network, I have a color in yellow, 2 sublobes. They could be really complex, they could be really simple, but yeah, they are those. Okay, so given an unrooted network, we say that the root is trapped if we cannot place it outside a cycle. What do I mean by this? Is I can place the root or the LSA anywhere here I want, but I cannot place it here because I will contradict in terms of the flow of time, because you will have edges pointing together. And in a cycle, I mean, if you consider this network as completely undirected, the root will be, you can go from the root and come back using different set of edges. So particularly, we say that the root is trapped in a 2 subblob if the root cannot be placed outside the 2 blob. Here I have an example. You can see these 2 blob with the nodes U and B. I can place the root here in this green node, and everything will be consistent. And I can place it here outside the blob, and everything will be consistent. But in this case, I cannot place the root outside this blob because then I cannot place it here because I'll have a contradiction. I cannot place it here, have a contradiction. So the only place I can place the root is inside that 2 blob. Okay, so we showed that if you have a network, you can replace any 2 subblob with an edge of a specific edge length. Here is the formal statement. You don't have to read it if you don't want to. But you can replace it with a 2 subblob with an adequate edge length, and then the concordance factor for these 2 blobs will be the same. So again, if you're using concordance factors, you cannot tell anything about 2 subblobs. In particularly, if the root is not trapped in a 2 subblob, then we know that that edge length must be positive. Okay, so now I can go, I need another definition. Also, it's quite technical. This is not how it's defined on the paper. So I'm simplifying it a little bit. So suppose you have a network, and then the display trees will be the trees you can obtain by looking at subsets of the edges that are consistent, and you can get like a species tree kind of thing, and then unrooting those. Again, this is a little bit informal, but think about how many trees I can get from this network, okay? So in this particular case, this network has to possible display trees. It doesn't matter which edge lengths I take from the hybridization events, I will get with one of those 2, end up. Okay, so Let me go back to no hybridization, so in a tree setting. So we say that a species tree is anomalous if the most probable gene tree does not match the unrooted species topology. So here you have a depiction of a species tree and a coalescent and gene tree generated. So here I have a silly example of the probability of a gene tree under the MSE is greater than the probability of the unrooted gene tree, which matches the unrooted species tree. So in the network setting, we say that, again, a little bit informal, a network will be anomalous under the NMSE if the gene tree, if there exists a gene tree that is not displayed, that is more probable than a gene tree that matches a display tree. So this was first seen by Claudia and Cecile, Soles Dumas and Anne, where they look at this network. This has one display tree, but the probability of that display tree for some set of parameters, numerical parameters, is less than the probability than the other gene trees. So for this particular network and some parameters, the most probable gene trees do not match the display tree. This particular network is called a tree-to cycle because you can see that it has, this is three edges coming together, and then there's two nodes, or two taxa, below the hybridization event. So, before I go to something else, when we consider 4 taxon binary semi-directed networks, there's 4 types of networks. So we say type 1 if it displays only one tree, type 2 if it displays 2 trees, and type 3 if it displays 3 trees. So networks of type 1 and type 2 are the only ones that can be anomalous, because if you are type 3, you are displaying all possible gene trees. So then there's no gene tree not displayed that has a greater probability. So the first thing we were able to show is that if you have a 4 taxon binary semi-directed network of type 1 or 2, if n contains a tree-to cycle, then there are some parameters such that under the NMSE, you will be anomalous. That is, let's look at an example. This is a 4 taxon semi-directed network. And you can see here in blue, I have highlighted a tree-to cycle like the one I just talked before. That one, I can assign some parameters to this network and find an anomalous network, or it will be an anomalous network. So as a partial converse, what we prove is that if you have a network that is anomalous and you assume that does not contain a hybridization event trapping the root, or if it traps the root, its removal will disconnect the network in a specific case, and this is the case I'm referring to. So you have a 2 blob trapping the root, and when I remove the 2 sub-blob, I have one taxon by itself and the other 3 together. In those cases, if your network is anomalous, then you know it must have a 3 sub 2 subnetwork. Now, what we conjecture is that if n is anomalous, then n contains a 3 sub 2 cycle. But we were not successful in proving that. It was really difficult. A lot of times I thought it was true. A lot of times I thought it was false. Yeah, I mean, it changed a lot. We thought we have really good directions of proof, but we were not able to prove that yet. So other things we do in this work, we present a new recursive algorithm to calculate concordance factors for any network. That's really nice. And we quantify the frequency of anomalous 4 taxon networks using simulation under a Verded hybridization process. And we generalize results for the extended coalescent model that Cecile just talked about. And if you missed that talk, you really missed on a great talk. So, yeah. So, yeah, thank you very much. That is it for me.
might note that trait evolution models are going to be central to understanding morphological diversity. There's a lot of morphological diversity out there, and they can be used to assess things like the tempo and mode of evolution, as well as poke at questions like selection and adaptation behind trait evolution. And with an increased ability to detect and infer reticulation, things like hybridization and gene flow, it's becoming increasingly recognized that these processes can play a large role in shaping patterns of trait evolution. And recently, there have been a lot of methods developed that are able to account for reticulate evolution, and that's what I want to talk to you today about, is a method that we developed that can account for reticulate evolution by looking at the various ways lineages can sort along a phylogenetic network. And so, when it comes to modeling trait evolution, the first thing we might want to note is that continuous traits are often going to be modeled with Brownian motion. This is also known as a random walk. And so when it comes to the distribution of species traits, we can think of this as a multivariate normal distribution with these parameters that represent the starting trait value, the evolutionary rate, and then also there's this covariance matrix that represents the shared evolutionary history between species. And so our model, I like to think of it as a hybrid of these two models here, this trait evolution on a network, and then also gene-level evolution. And so first I'll talk a little bit about these approaches and then talk about how it's kind of a hybrid of these two. So first, we want to look at this Bastide et al. Model, which is trait evolution on a network. And so what it does is it accounts for gene flow by directly using the network to compute those covariances. And the way it does this is it considers all of the different paths that lineages can travel along that reticulate history, and it asks what are the covariances along these paths, and then it weights those according to the inheritance proportions noted as gamma and 1 minus gamma that represent the proportion of genetic material that each parental lineage donates to the hybrid. And that's how we get these weightings here. And so the covariance overall is just going to be a weighted average along each path. And then there's this little asterisk here where we have reduced variance in hybrid descendants. And I'll talk a little bit more about that when we talk about the results. However, one thing to note is that it's going to assume that lineages are sorting independently at a hybridization event. So when it comes to going left or right, it's going to assume that they are both going to be moving independently of one another. And this might not always be the case. So if we think about things like incomplete lineage sorting or the coalescent, some lineages might coalesce prior to that hybridization event, and they're no longer independent of one another. And this can actually affect these weightings. And so one way that people can account for this discordance in processes like incomplete lineage sorting is by using this quantitative trait model from Gibbons et al. And what it does is it uses gene-level histories to directly account for that discordance. So if the gene tree histories are in purple here, essentially what it does is it takes an average of the covariances from each of those gene trees to compute the overall covariance. And so I like to think of our model as a hybrid of these two in the way that it considers the lineage sortings or the different paths that lineages can take along a reticulate history. It uses those lineage sortings and covariances at the species level. However, they're going to be weighted by the lineage sortings found at the gene level. And when I say weighted by the lineage sortings found at the gene level is we can look at each gene tree and we can give it a lineage sorting based on whether the gene tree histories, the lineages go left or right. So for example, these two gene trees here both have a lineage sorting of H2 because they both go left at that hybridization node. And so what we can do is we can count up all of the genes that have a given lineage sorting, and then we can use those as our weights. And we just say, what's the proportion of genes with a given lineage sorting? And we can use those as our weights when we are computing covariances. And one thing we can note is that the multi-species network coalescent can make predictions actually about these lineage sorting weights. So if we think of the multi-species network coalescent as a distribution for our gene tree histories, in that it's going to be primarily controlled by the effective population size, where this effective population size on average is going to determine whether lineages coalesce or whether they do not prior to a hybridization node, is that's going to ultimately affect the weightings that we might predict for each of these lineage sortings. And so one thing we can do is we can form different hypotheses by using different weighting schemes for each of these lineage sortings. So for example, we might have the weightings predicted from this multi-species network coalescence. We might also have just what we would observe directly from the gene trees themselves, or we can also incorporate other information such as gene ontology. So for example, if we're interested in a trait like tooth size or canine size, we might hypothesize that genes related to carnivory might play an important role in shaping tooth size. So we might want to weight things accordingly based on all of the genes that we find important to carnivory. And we can develop different weighting schemes based on that to test various hypotheses. And so we have this model, and now what we're going to do is we're going to compare it to other trait evolution models and kind of assess the performance. So what we did is we conducted a simulation study to assess model performance, where we used a variety of simulation conditions. I'm only going to be talking to you today about two, when we simulated under the network model, the Bastide model, and then under our lineage sorting model using weights from the multi-species network coalescent. And we did this under a variety of topologies. I'm primarily only going to be talking to you about this H1 network here, which has one reticulation event. And so for our network model simulations, what we did is we varied the inheritance proportions, and then we kept our evolutionary rate the same, and then we asked, what is the evolutionary rate when we fit a variety of models? We have the major tree model, which essentially is if we get rid of these dark blue lines here, and then we fit the resulting tree. We have the network, which is the true model, and then we have our lineage sorting models, which is when we're assuming a small population size, a large population size, and then something in the middle. And what we can note is that if we fit an incorrect model, is that the evolutionary rate is actually going to be underestimated. So the network was able to recover the true evolutionary rate on average, whereas everything else tended to underestimate it, regardless of the inheritance proportion that we used. And we believe that this underestimation is primarily due to that reduced variance feature of hybrid descendants. So if we look at the covariance matrix of the network model, of the true model, that reduced variance property, we can note along the diagonal that there's this slightly lighter red. That's that reduced variance property. And we can actually compare this to the worst performing model here, which is the lineage sorting model that assumes a really high population size. The only difference between these two models is actually in these 4 cells right here. This does not have that reduced variance property, but otherwise the covariance matrices are exactly the same. Another thing we can note is that for the tree model, it was actually the least biased. And this can be kind of a comforting result because we sometimes use tree-based comparative methods, even though we assume that there's an underlying network describing the evolution. However, we might want to temper that comfort a little bit because this seems to be context dependent. The tree-based model actually ended up being the most biased on other topologies that we tested. Another thing we can note is that for relatively high inheritance proportions, so as the network becomes increasingly tree-like, regardless of whether we use the incorrect model, the amount of bias decreased. So we're a little bit more robust to model misspecification at high inheritance proportions. And so this can lead us to conclude that tree models might be adequate under certain conditions, specifically for relatively high inheritance proportions and for specific topologies. And this can be helpful because not all comparative methods can accommodate reticulation, and we, as I said, we can't always have a network to fit these models on. And so, for example, in this study here where folks were looking at monitor lizards, they fit a variety of tree and network-based models, and I would argue that their tree-based trait evolution models or their conclusions are still reliable because for the network that they inferred a relatively high inheritance proportion. And then, so moving on to our simulation model where we simulated using our lineage sorting model, using weights from the multi-species network coalescent. Here, we kept the inheritance proportions the same, but we varied the effective population size. And then we fit all of the same models. However, in this case, our true model is using the actual effective population size. And so what we can note is that there are biases whenever we don't account for effective population size. So the two models that don't even consider effective population size are the network model and the tree model, and they always tended to overestimate the evolutionary rate. However, their estimates became much more biased when the population size was large. Additionally, when we assume a relatively large population size, when the true population size is small, that led to biases, as well as assuming a really small population size when the true population size was large, also led to biases in our estimates of evolutionary rate. Another thing we can note is that for relatively small effective population sizes, the overall bias, or when we use a misspecified model, was relatively small. And this can be kind of a helpful result because in cases of hybrid speciation or adaptive radiation, the system itself is going to tend to have inherently a small effective population size. So even if we misspecify the model in these cases, we might be okay in terms of our overall results, even if we misspecify the model. And so in conclusion, we have this comparative method that can account for the various ways that lineages can sort along a phylogenetic network. And this is a relatively flexible framework that can be used to test a variety of evolutionary hypotheses. And it's currently implemented in Julia as a fork of phylo networks. And it is available if you scan this QR code, or you can see it on my GitHub, JJustison. I'd like to thank the members of the Heath Lab, my graduate committee, and then also my funding sources. And thank you. If you have any questions, feel free to contact me at my email, or if you are looking for postdocs. Thank you.
Good afternoon, everybody. Thanks for being here. So I'm going to talk about an empirical example of a network. And so and I'm going to talk about a microbe, a very famous microbe. So Saccharomyces cerevisiae is a well-known model organism. I'm sure you're going to hear about it many times during this meeting. And it's also associated with a lot of multiple human activities. For example, it is a predominating yeast species in processes like brewing, baking, and winemaking. And because of this tight association with humans, is considered a domesticated species. And so the general, this is how we typically represent the relationship between the different strains. And so this neighbor joining tree generally reflects the geographical and ecological origin of these strains. And for example, here, I think it's better. I know we don't have the. Well, here in these very long branches, we have very divergent strains that were isolated from natural forests in China and other regions of East Asia. And so the point here is, so Saccharomyces cerevisiae is a domesticated species, but is not exclusively a domesticated species, okay? So there are strains that are associated with completely wild environments. And so even though it's a very famous model organism, we don't know much about actually the evolutionary history of this species. So one of the questions that we don't know about is how many domestication events are behind the domestication history of Saccharomyces cerevisiae. And so to address this question, we have a student in the lab that collected more than 3,000 strains from all over the world. So he collected the sequences that are available, and he performed SNP calling pipeline for all these strains. And then he performed this neighbor joining tree. And so, for example, you can see that in on your right side of the tree, you have these very long branches here. So this blue cluster forms a major cluster that includes all the wild strains. And then we have other major clades that are here represented in the middle that are domesticated clades involved in fermentation processes. We have another clade here that represents the beer strains. And then we have finally this very large clade that includes all the strains from Saccharomyces cerevisiae that are involved in wine production. So within each of these major clades, we have subclades, okay? And so we based on this general structure to define the different groups of strains. And so we looked at the patterns of diversity and between and within these different clades. And one thing that stands out was that the Asian fermentation strains and the Asian wild strains, they are equally divergent from all the other known Asian strains, and they are more closely related between each other than they are from the other ones. And also, among the least diverse strains, we have the wine-related strains. So we have a clear sign of reduction in population size due to the domestication event. And so we went also to compute the allele sharing between the different combinations of clades. So I think we already heard talking about many times about the d-statistics. So I just quickly, if we have a d-statistics close to 0, it means that we have no introgression from population 3 into population 2. And we have, if we have a d-statistics larger than 0, then we have an evidence for gene exchange between P3 and P2. So here on the heat map, we have the P3 in vertical, and in the bottom we have the P2. And you see that for many of these entries, we have significant values of these statistics, giving us the idea that these strains, they have been exchanging quite a lot of DNA. And so the point is here, and what we really want to address are these patterns compatible with the 1 or 2 domestication events? So with that purpose, we focus on these 3 main clusters. So the wild cluster, the European cluster that includes all the wine strains, and the Asian fermentation clade. And we used a coalescent framework based on site frequency spectrum. So we built up some models to test these different hypotheses. So in the case of a one domestication model that I have here, I basically modeled the evolutionary history of Asian domesticated and European domesticated clades as a hierarchical continental island model. So these models are known to capture the features of structured metapopulations where you have these little islands that exchange genes backwards in time to the continent, and not on the other way around. And so in the case of one domestication event, we have a first split from the East Asian wild populations and the ancestors of the domesticated species, of domesticated strains, followed by the divergence between these two groups. And so we tested this model together with the models including 2 domestication events, one involving an older split of the European strains, and the later split between the Asian strains, the domesticated and the wild, and the third model where we have an earlier split from the Asian domesticated strains, followed by a split between the European domesticated and the East Asian wild strains. And so just to give you a little bit more details about the modeling, so Saccharomyces cerevisiae, it's a facultative sexual organism, so the life cycle includes parts with clonal reproduction, and others with selfing, and every now and then they do outcross. And so to take that into account in the model, we basically represented each population by a sexual component and an asexual component. So in the sexual component, coalescent events should happen at a faster rate, and in the asexual component, we should not have any coalescent event. And so the lineages, they have a given probability of migrating from the sexual component to the sexual, and then with the least probability to the outcrossing component. And so one thing that we did was to also try models where these values, these transition rates were fixed. So they were the same for the Asian domesticated measured clade and for the European measured clade, and we tested models where these transition rates were different. And so here we have the topologies, and in the bottom I'm showing you the likelihood, the composite likelihood estimates. So this was done with all the data sets. So we have around 26,000 SNPs to do this analysis. And in the bottom, I'm showing you the likelihood computed based on one SNP per 500 base pair windows, 1,000 base pairs apart, to decrease or remove to the max the effect of linkage disequilibrium, because we need the likelihoods to be, the composite likelihood to be close from a real likelihood to be able to compare formally the models with the archaic information criteria. And so when we look at the three models, we see that is the model with the two domestication events with an older split from the European clades that have the highest support. And then if we compare them with models with migration, then our likelihood improves. And finally, if we take into account group-specific sexual and asexual transition rates, we have a much better likelihood. And from all these 9 models, we have better support for a model with two domestication events with an earlier split from the European population. So that's the take-home message I want you to keep. The patterns of genetic diversity in Saccharomyces cerevisiae are compatible with two domestication events with an earlier split from the European domesticated strains, and we have clearly evidence that these strains have exchanged genes since they split, and so we should no longer think about microbes as a tree, but rather as a network. And so I will be happy to take any questions.
Nice talk. Since these genes are domesticated in really distant regions, so what's your hypothesis of how they exchange genes? Like exchange of strains between the cultures?
Yes, I think that's very speculative, of course, because we don't have any written proof of that, but it's probably linked to just the exchange of strains between different producers from different regions in the world.
Thank you, that was fun. I have 2 questions. The first one is, the whole analysis is based on SNP comparison, on sequence comparison. If you look at gene content, what do you see, and would that lead you to a different tree and therefore maybe different implied hybridization events?
If I'm understanding correctly the question, in terms of gene content, it reproduces mostly the picture I showed. So what we see with SNPs, we see with other type of structural variation or other kind of data.
And then my second question, do I have time for one more? Is, so these are domesticated strains.
Yes.
Where you might expect to see some convergent evolution, because do you think that's playing a role?
That's a possibility. When I saw the results from these statistics, for example, I thought, okay, wow, that's quite a lot of exchange. So either we have some groups of strains that basically they just, you just take one strain, two, and then you put them together, and then your process gets better, and you keep reproducing these hybrids. Or we have convergence evolution. I looked quickly at the distribution of these statistics across the chromosomes, and it's more compatible with the general process, so really mixing two types of strains together to produce something than convergence evolution, because it's not specific from a particular region of the genome. But that's very preliminary still.
Thank you very much.
Thank you.
Hello, everybody. Thank you very much for being here this afternoon. I'm Kevin Kong. I'm a postdoc at the University of Wisconsin-Madison, working with Claudia Solis-Lemus. Today, I'm going to talk about some of the computational improvements, computational update that we have made on a popular network inference method called SNAC, and then we're going to call it SNAC2. It's still in the progress of developing in order to improve its scalability or to improve the speed of the inference. I'd like to start off my presentation with a listing number of existing computational tools to infer phylogenetic networks. And this is because we have been working on a review paper, and I'm impressed how network methods have developed in the recent past. We have Phylonet, which is a gigantic program that has multiple functions using multiple optimality criteria like parsimony, likelihood, composite likelihood, or pseudo likelihood, and Bayesian using gene trees, sequence alignment, or biallelic size. We have a couple good composite likelihood methods called SNAC and Finest, which uses quartet concordance factor for SNAC and quartet side pattern frequencies for Finest. We have some packages in BIS2 framework using Bayesian. And we have also some innovative methods that combines likelihood and multiple statistical tests in Nanook, or RFNet2 NetRocs that are also fast, which also have little different biological assumptions. And all of these methods that I discussed are focusing on hybridization. So reticulation in the network inferred in these programs will represent hybridization. But we're trying to infer other non-tree-like processes like polyploidization that are implemented in other softwares. And today we're going to focus on SNAC here. And to briefly talk about how SNAC works, it is an acronym for Species Network Applying Quartets. It's a composite likelihood method where the idea is instead of tackling the big difficult problem that may be computationally very heavy, we're breaking the problem down into smaller pieces. And we find a solution for those smaller pieces and combine them together to approximate the likelihood for the bigger problem. So smaller pieces here represents taxon set that compose of 4 species. And there are n choose 4 number of this selection of 4 species, which means it increases crazy as you have more number of tips because n represents the number of leaves. And given 4 taxa at a time, let's say the 4 taxa we selected are species A, B, and C, and D. As Hector mentioned, there are 3 ways to find the relationship among these 4 species when unrooted, where A and B can form a clade, or A and C can form a clade, or A and D can form a clade. And the true probability of each of these unrooted quartets under the network multi-species coalescence can be computed. And we're going to call that quartet concordance factors. And observed quartet concordance factors can be computed. We're going to call that X by counting the gene tree, number of gene trees that matches with each of these quartet topologies. So SNAC seems to have some ingredients that are supposed to be fast, but is it actually fast? So these 4 plots represents the computational time using existing version of SNAC, where the x-axis represents the data set size in terms of a number of gene trees, y-axis computational time measured in hours. The top row represents the network topology with 10 leaves, the first one with 1 reticulation. The first one with 2 reticulation, the second one with 3 reticulation. The bottom 2 plots represents a little bigger network with 20 tips with 1 reticulation and 3 reticulation. When the network size is relatively small, the top rows, the computational time is definitely good. It's taking about half an hour to finish the analysis. And even though it has 3 reticulations, it finished the analysis in average about 1 hour and a half. But when we have larger network, not even larger, 20 leaves with a single reticulation, it takes 24 hours to finish, and we increase the complexity by adding 2 more reticulations. It took 48 hours or more to finish the analysis. Well, 2 days for a network analysis doesn't seem like a big number, but compared to the tree analysis, we definitely need to improve the speed of the network analysis. So this logically leads to my question, can we improve the scalability of existing SNAC? And I tackled this question by using 3 strategies, by implementing 3 strategies. 2 of them are trying to speed up the composite likelihood calculation, and 1 of them trying to make smart moves during the network search. And one way to speed up the composite likelihood calculation is very straightforward. Because the first version of SNAC uses a single thread, we just use multi-threading. So to make this a little bit more simpler, let's say these workers on the left side has a task to compute a quartet likelihood. If you have a single thread, only 1 worker can go into core and they can compute likelihood, and then we have to wait. Next person has to wait the previous person to finish, and then on and on. And this is quite an efficient way of using computational resource. So in SNAC 2, we multi-thread this process. So multiple workers can go into computer core at the same time. Therefore, more people can be happy, and the analysis is supposed to be much faster. So we're pretty sure this should speed up the process. The second strategy to improve the speed of the composite likelihood calculation is by subsetting using a portion of quartets. So let's say these are all of the quartets that we extracted from the bigger networks. And the idea is let's use only a portion of that. What happens if you use a portion of that? And we're going to call that prop quartets, and this can be set in the next version of SNAC. If we set prop quartets as 0.75, we're going to use only 75% of the extracted quartets. This might seem unintuitive because if we are using only a portion of the data, this will obviously lead to the decreased accuracy. But it might not be the case because when we have tips, especially when we have multiple individuals for a single species, the relationship between species are not concentrated in a single quartet, but they appear in multiple different quartets. So if we want to know the relationship between A and B, they're occurring in multiple quartets, and even though we ignore some of them, we still can know the relation. We hope we can still recover the relationship between A and B as long as they have very strong signal. And the last thing that I try to do is to make a smarter move during the network search. So during the network search, what's happening is we have a network topology, and we randomly select one of those topologies to make a move or make a small alteration on the topology, and we compute the we compare the likelihood of those 2 topologies, and we decide either we want to make a move or not. So the idea here is the edge to make a move is randomly chosen. And in our version of, so the next SNAC 2, we are trying to make more informed selection of the edge. So the edge to make move can be selected based on the weight, where the weight is simply the sum of absolute difference between expected concordance factor and the observed concordance factor. So the idea is if the weight is large, that means there is a big discrepancy between the expected concordance factor and the observed concordance factor, which probably means this edge might not be a good representation or good fit for the data. And this potentially avoids to make a change in a topology that is already good. And if we just randomly select an edge and make a change, the resulting topology will have worse composite likelihood, and we step back. By removing that unnecessary step, we are expecting to improve computational efficiency. So I try to see if combination of these three strategies will improve the speed of the analysis. And here is the simple pipeline for our simulation study. I manually selected 4 networks, again with 10 tips with 1 and 3 reticulations, and 20 tips with 1 and 3 reticulations. And we simulated a set of gene trees ranging from 300 to 3,000 using phylocore simulations. And for each of those gene trees, we simulated sequence using MS under HKY model, 1,000 base per loci. And using the simulated sequence alignment, we estimated a set of gene trees using IQ tree. And using those estimated set of gene trees, we estimated network using different computing powers, number of processors ranging 4, 8, and 16, and we gave the true number of hybridizations to make things easier. We use SNAG with default settings, and we use SNAG2 using probe quartets ranging from 0.5 to 1.0, which means we're trying to use half of the quartet or all of the quartets, and probe QR ranging from 0 to 1, fully random for network searching to fully guided network searching. And we evaluated their performance by measuring runtime and accuracy. And accuracy, I mean comparing the true network topology and the estimated network topology using hardwired cluster distance. So here is the plot for the accuracy. X-axis represents the number of processors, and Y-axis represents the topological error. So as the value gets closer to the x-axis, that means the estimation is getting more accurate. The plot, there are 3 sections. The plot that has red outline is the result from SNAG1, and 4 blue violin plots are results from SNAG2. And as you move among the 4 violin plots from SNAG2, as you move left to right, as the color gets darker, that means we're using more quartets for its computation. As expected, the accuracy did not change. And this is good because we are expecting SNAG2's accuracy to be same as SNAG1. So this is actually a good news. But what we see here is when the number of processors is 8 and only half of the quartet was used, the accuracy was actually worse than when more quartets were used. And this is exactly how we expected. So even though we didn't have perfect estimation, I was quite happy with this plot. And this same pattern was observed even though when we had, so each of the rows are different number of gene trees used as input, on top 300, on the bottom 3,000. The same pattern was observed as SNAG 1 and 2 had very similar accuracy. And if you use half of the quartet, the accuracy declined. But if you use 70% or 90% of the quartet, the accuracy did not change much. A more interesting story, which is efficiency here. So again, x-axis represents the computing power, y-axis represents the running time, and things are getting faster as it gets closer to x-axis. And likewise, red SNAG1, blue SNAG2, as the color gets darker, more quartets are used. As the number of processors increase, we can see the computational time is getting faster. And explanation for this pattern is because we are more efficiently using the computing power through multithreading. Another thing is, as we use more quartets that are extracted from the network, computational time increased, which means if we use less number of quartets, things will be faster. But we don't want to use half of them because it's going to result in incorrect topology. But 70 to 90% might be a good number. One thing that I need to think a little more harder is the effect of probe QR. Like I couldn't find some clear pattern of using this guided network searching. So this is my next step to see what are the patterns that we observe. Is it going to increase accuracy, or maybe it'll decrease accuracy because it might lead to becoming stuck at the local optimum. So my next step is to understand the relationship between probe QR and the accuracy and efficiency, and finding good ways to identify the best setting for probe quartets and probe QR given the data. This is for the empiricists who want to use this method, and also we want to try the analysis in various empirical data sets. Thank you very much for listening, and we have 3 other very interesting network-related talks coming up from our group. We have Nathan Colville right next to me. He's going to talk about network merging. We have a poster working with Lydia Molly and Emma Lemberg. Very interesting. We're going to talk about the trait evolution of the network, and also Claudia Solis-Lams on the state-of-art inference method for complex phylogenetic networks. Thank you very much. Yes.
I missed how you select the quartets when you use a subset of all possible. Is that just random, or?
Yeah. So if the selection is biased towards some taxon, that's going to be not good. That's what we thought. So we are making selection randomly. Yes. Yes, yes, that's actually a very great idea, and then. Right. Yeah. Actually, I'm trying to think about a good way to sample those quartets. Like when we are trying to sample a subset of that, of course, the easiest way is sampling it randomly. That's what we have. And then once this seems to be a good way to move on, then definitely that might be a great way to implement. Thank you very much.